All right. Have a seat, please. It's time to start the afternoon session. Actually, the session 5, the new pathways with potential impact in the treatment of early breast cancer. I'm chairing, my name is Mark Toy, Kyoto University, and the co-chair.
I'm Zefei Jiang from Beijing, China, and it's an honor here to chair this section.
I think that this session would be very exciting. The first speaker would be Andrew Tutt, the Institute of Cancer Research, King's College London. And he is going to talk about the targeting the DNA repair. Andy, please.
Thank you very much. A great pleasure to be asked to speak at the St. Gallen conference. Thank you to the chairs. So I'm going to be discussing the targeting or the potential for targeting DNA repair. And I'd like to start really with the evidences that there is a DNA repair defect to target within breast cancer. And so what I've illustrated on the left here is that breast cancer is a disease really typified by chromosomal instability rather than sequence changes, SNVs, which other forms of cancer have as the dominant form of mutation. Breast cancer, rather like ovarian cancer, is dominated by major chromosomal structural changes, amplifications, gains and losses. And sequencing analyses shown on the right have demonstrated that there are both SNV signatures, mutational signatures, but also rearrangement signatures, which again will be driven by major breaks in DNA and their repair. And some of those processes involve particular DNA repair pathways controlled by BRCA1, BRCA2, and other members of a process called homologous recombination. Now, deficiency in this process has had rather a lot of attention. And that attention is because this is a very important pathway for accurately repairing DNA double-strand breaks. And these happen a lot in tumor cells that are dividing rapidly, and that genes we know predispose to breast cancer, like BRCA1 and 2, but several other predisposition genes act within homologous recombination repair. When it is deficient, what I'm showing on the right is that this can leave a pathognomonic mutational scar or series of tattoos on the genome of that cancer. And so investigators have now started to try and study whether studying that genome and the tattoos or scars on it can help identify tumors that have that DNA repair defect. And we're really asking, how could we target that? These analyses, such as the HRDetect analysis, requires a combination approach. So it's not just looking at one tattoo on the genome, it's looking at a combination of tattoos and building up a picture in order to get high performance of the assay. And that requires that you can see the whole genome sequence, and that has to be done at high depth, and that's challenging to do on formalin-fixed materials. So this is a developing art at present. However, the preliminary data do suggest that there's significant promise in identifying a wider group of cancers that have deficiencies in this important DNA repair process. It's important that damage happens to both copies of these homologous recombination genes, so both copies of BRCA1 and 2 are damaged, either one in the germline and one somatically or both somatically. You can have a methylation event that causes an HRD scar. So BRCA1 being turned down by methylation can also cause a deficiency. And there are a number of unexplained causes of deficiency that we need to try to identify, because this may reveal patients who we can target. So the poster child for targeting a DNA repair abnormality is the targeting of PARP and synthetic lethality with loss of function for the BRCA genes. Poly-ADP ribose polymerase is shown here. It has a catalytic domain on the right, and on the left, my left, a zinc finger binding domain. This works by binding DNA at the sites of single-stranded damage, and then as you go around this circle, you see that PARP is creating a polymer, and this attracts in DNA repair factors that allow the repair. But what now seems crucial is that the drugs we're using to inhibit PARP actually trap that PARP onto the DNA, and this seems to be important for the way that they interact with deficiency of DNA repair. So whether you're trapping PARP on DNA or whether you're sticking a piece of platinum on DNA, you arrest DNA replication forks, and this is where the loss of function of BRCA1 and 2 become important. Because if the tumor cell doesn't have this working, it can't protect its DNA replication fork and engage HR repair, and that tumor cell will die. Whereas the normal cell has functioning BRCA, can protect its replication forks, and is more likely to survive. So these two approaches target similar biology. Now, PARP inhibitors have achieved marketing approval in patients with germline mutations in BRCA1 and 2 in an advanced disease setting and are beginning to be looked at in early disease. And this applies to two drugs, talazopanib and to olaparib. These trials compared with standard of care chemotherapies, but importantly didn't have a platinum comparator because it wasn't considered a standard of care. But prior platinum exposure was allowed as long as there was a significant disease-free interval. And these data you will have seen, I'm sure, many times. These are for talazopanib, showing meaningful improvements in progression-free survival and, importantly, delay of deterioration of quality of life. These are the data for olaparib for progression-free survival. But even in this very defined biological situation, we clearly still have a significant problem. These patients are all developing progression, they have metastatic disease, and this is happening early for some and not at a long delay for others. So resistance is an issue. There is much still to do. And it may well be that the use of platinums, while very useful, is also leading to the selection of patients who are inherently resistant. We've shown, or my colleagues in the TNT group have shown, that platinum is also a highly effective approach, and that's also leading to its consideration or its use broadly in early forms of the disease. And so with the work of a number of groups looking at platinums in neoadjuvant therapy, on the left, Gephardt-Sixto, on the right, the CLGB study, we know that the addition of platinum in triple-negative breast cancer in a neoadjuvant setting leads to an increase in the pathological complete response rate in these two studies. Now, if you look within the BRCA mutation population in Gephardt-Sixto, you can see here in blue that this population already seemed to do rather better with the anthracycline taxane regimen used than the non-BRCA carrying population. The difference by the addition of a platinum is therefore smaller, more difficult to show. These patients with naive tumors actually respond very well to the DNA-damaging, DNA-interacting drugs, the part of our standard of care, cyclophosphamide being one of those. It has been looked at as to whether adding in PARP inhibitors to platinums in the neoadjuvant setting can add value. So this was the I-SPY2 program, pulling out the triple-negative population as those who benefited from the addition of veliparib, a catalytic PARP inhibitor, with carboplatin in the triple-negative population. And then another trial, Brightness, very carefully and elegantly dissected what the role of each of those new agents there was, carboplatin or veliparib. So this is the combination of the two, and this is with a placebo control, just looking at carboplatin or the standard of care regimen. And essentially, what that showed was that the benefit for increased response was as a result of the platinum component, not as a result of the addition of veliparib to platinum. When you look at the BRCA carrier subpopulation, there was nothing to suggest the effect was restricted to the carriers. And in fact, you can see that the carriers here have a very high response rate to the standard of care. Let's get that to come up there. So being a carrier means you have a higher baseline response rate to the standard of care treatments, as I showed you before. So clearly a wider population than the carriers benefiting from the addition of a platinum. So what's going on with the PARP inhibitor effect here? And this is complicated to explain, but it's clear that veliparib is a little different. I've tried to review this in a manuscript, the citation of which is there, but essentially this is about trapping or the lack of trapping of PARP with some of these agents. It seems very important that you trap PARP onto DNA in order to elicit this synthetic lethal effect and an effect on the tumour. And there are differences between the different PARP inhibitors in their trapping ability. So you see here that veliparib is a very poor trapper, whereas a drug like talazopanib is a very potent trapper, as in fact is olaparib. So do we have data about the potent trappers in early breast cancer? So in the last year, this study, a Neo-Adjuvant study from Jennifer Litton and colleagues, has looked at this. And these patients were treated solely with talazopanib for 4 cycles prior to surgery in a neoadjuvant setting. Again, these are all BRCA mutation carriers. A small study, the endpoint was pathological response. And just with a trapping PARP inhibitor, a pathological complete response rate of over 50%, over 60% if you include those patients with very minimal residual disease, could be achieved with manageable toxicity. So this is going forward into a much larger study to examine these drugs in this context. But how do we find if there's a broader population that may have HR deficiency targetable by PARP inhibition? And that's going to take studying tissue. And this is a study led by Nick called RIO, which is in a pre-neoadjuvant or pre-surgical study in which patients who have sporadic triple-negative disease and a control population with BRCA mutation are exposed to a potent trapping PARP inhibitor, rucaparib, for 2 weeks, and then they have biopsies before and after, ctDNA before and after, and changes in proliferation, but also in ctDNA markers can be analyzed in those patients against markers that may indicate HR deficiency, such as the scars or tattoos of HR deficiency, and some new biomarkers that measure the function of the recombination process by measuring RAD51 foci, which has recently become a more validated method. So there are a number of studies now that are developing approaches for platinum or PARP inhibitors in an early breast cancer setting. They're listed here, and I don't have time to go through all of them, but I'm going to focus on just 2 for a little more discussion. One is GEPAR-OLA, and the other is the Olympia trial. So GEPAR-OLA is a study, neoadjuvant setting, combining either olaparib or platinum with standard of care sequential neoadjuvant therapy with an endpoint of pathological response. And that will give some signal as to the relative effects of these drugs. But it's also really important that we study those patients who aren't doing so well at the end of those treatments. Not everyone has a pathological complete response. Perhaps 20 or 30 patients have some significant disease, and they're anticipated to have worse survival. So if we focus on these patients, we can try and understand what mechanisms of resistance are at play and which are the more prevalent and which are the same and overlapping between these drugs and which are different. And that may help us in interpreting trials like Olympia, where patients who have residual disease after neoadjuvant chemotherapy are randomized to receive olaparib. And we want to try and understand who benefits best from those situations, and the development of resistance to these drugs will be important to analyze that. Resistance can develop through a number of mechanisms, and one mechanism that's been found out is reversion mutations, ways in which the BRCA gene gets fixed. And so tumor cells with BRCA defects are operating with a short gene and a short gene product, short protein. But the DNA repair defect in these cells can bizarrely cause the break site, or sorry, the mutation site, to get repaired. And that leads to a normal or a near normal length protein that functions again. So you then have functional HR and PARP inhibitor resistance. This is expected to cause resistance to both platinum and PARP inhibitors and may predict the likelihood of benefiting from one drug after you've had the other. So we need to know the frequency with which this happens in the clinic. Does it apply to other HR genes? And how could we delay the emergence of these effects and treat the fixed BRCA tumors? Now, you can track this as a patient goes through treatment, at least in a research setting, so this is from Nick's group and his ctDNA analysis laboratory, looking at patients as they go through their treatment journey on platinums and PARP inhibitors. And this study could show that the BRCA locus could get reversion mutations of many different types in the same patient, and you could track these developing as the patient developed resistance and exposure to the treatment. So potentially, one could use this to monitor treatment. Now, some resistance mechanisms are not cross-resistant between these drugs. So my colleague in our center, Chris Lord, has published work showing that PARP, which normally binds to DNA, here's the DNA here, here's the PARP, needs to bind to trap. Mutations can develop which stop that trapping, and that leads to PARP inhibitor resistance. But it would not be predicted to cause platinum resistance, and that might affect the choice you make for the next treatment. Another alternative is that you lose a component of something called the shielding complex. That can re-enable the recombination within the tumor, make it resistant to PARP, but that would actually make it phenomenally sensitive to a platinum. So knowing which form of resistance your patient might have developed may be really very important. And we can model this and test these hypotheses now in patient-derived models. So we're now making models from patients who've perhaps got PARP inhibitor-sensitive disease, platinum-sensitive disease, you see at the top, and their PDX model is PARP inhibitor-sensitive. At the bottom, this patient has platinum-resistant disease, and the PDX from their tumor was also PARP inhibitor-resistant. We can take candidates we know from the lab are potential resistance mechanisms, we can knock them into a sensitive tumor, and we can validate that that actually causes resistance. So this is a real potential mechanism, and try and understand how frequently that happens in the clinic, and we can build the cellular models, patient-derived organoids, to further validate these. So what might this mean? The potential impact of this could be that in the future you've treated a patient with a platinum-containing neoadjuvant regimen, or perhaps a PARP inhibitor neoadjuvant regimen. And at the end of that, this is one of the relatively smaller number of patients who've got significant residual disease. And you might then be able to test their tumor or their plasma and determine whether they had a BRCA reversion or fix-it mutation, or another different cause of PARP inhibitor resistance. And this might drive a different decision. So for a reversion mutation, one might be able to predict they're not going to respond to a follow-on PARP inhibitor or re-exposure to a platinum. But on the lower situation, they may have had a PARP inhibitor, but they may be about to have a really exquisite response to a platinum. So these are going to be important things to try and understand in the future. Can we drug the mechanisms by which these tumors are resistant to these DNA repair targeted therapies? So we know that the ATR kinase is really important for protecting these DNA replication forks against the causes of their arrest. And these are drugs that are in the clinic, and when you combine them with agents like platinums or PARP inhibitors, you can substantially increase the effect on models like the PDX models that I was showing you before. Sorry, that's overdone there. And these are synergizing with trapped PARP or platinum. So there are trials now addressing these combinations, admittedly in advanced disease, and looking at their effect across triple-negative breast cancer. So this is a combination, so this is olaparib alone and olaparib with an ATR inhibitor. And importantly, not just looking in those with BRCA mutation, but those with mutations in other HR genes and those with sporadic, more normal triple-negative disease. So we'll get understanding of the signals going on there. It also seems that targeting DNA repair and possibly increasing replication stress, replication arrest, and genome instability may inflame tumors that are previously cold, something we would like to do to exploit immunotherapy. And this may happen because, as I say, replication stress is induced, and that causes cytosolic DNA release and a cGAS sting response. It may cause more immunogenic forms of cell death, or it may increase tumor mutational burden. This is all quite speculative at the moment, but trials are now looking at combinations combinations of these agents with immunotherapy, such as Tapatio. And here at Mediola, you'll see a trial which is not just focusing on mutation carriers, but also, again, those with HR mutations in other genes or those with sporadic triple-negative breast cancer, and examining combinations of PARP inhibitors and checkpoint inhibition. So much we will learn. So, in conclusion, HR deficiency is significant, both in familial breast cancer and in sporadic breast cancers, perhaps enriched in those with triple-negative disease. Targeting this is now approved with 2 drugs in the advanced disease setting in those with germline mutation. The role in adjuvant therapy will be answered by trials like Olympia, which is due to close in a couple of weeks' time. The analysis of HRD by biomarkers is in development. There are a number of genome tattoo scar assays, functional assays like RAD51 in development. DNA repair deficiency is not static, it can be re-enabled, and that will affect potential response to follow-on drugs after platinums like PARP inhibitors. So we need to study these different resistance mechanisms and what they mean for follow-on therapies. There are ways of drugging resistance that are being explored, and combination approaches with immunotherapy have a sound rationale, and hopefully we'll hear more in the future. And I thank you very much for listening to me, and also acknowledge many colleagues and our funders, Breast Cancer Now and Cancer Research UK. Thank you.
Thank you very much. Excellent talk. Any question? Can I start with you showed about the resistance mechanism, and somehow the platinum resistance and the PARP inhibitor resistance may be overlapped, or how it is overlapped, or?
I think for the reversion mutations, these mutations that can fix the gene, they do overlap, and I think that's probably driving quite a bit of what we see of the lower response to PARP inhibitors in patients who've recently progressed on a platinum. That probably explains a significant proportion of that. But what I'm trying to point out is that that's not all of the ways resistance can happen, and there may be some ways in which you would still really expect a good platinum response after a PARP inhibitor, or potentially a PARP inhibitor response after a platinum, because platinum resistance can happen for many reasons. So we need, I think, to care to study this well in order to inform next therapy. And we have the hypotheses from the lab. We can now test them in the major trial correlative biology programs.
The additional question, in the presence of neoadjuvant chemotherapy, in terms of the resistance mechanism, how the neoadjuvant chemotherapy affect on the...
Sorry, it is terribly difficult to hear up here.
Oh, how the neoadjuvant chemotherapy, how it affects on the resistance mechanism. I mean, before the neoadjuvant or after the neoadjuvant,
yeah,
the resistance mechanism may be changed or modified?
Yes. So I think the neoadjuvant setting gives us the opportunity to study it in a way that we can't study it in the adjuvant setting, because there's nothing to go back and look at except ctDNA possibly. And so I think learning from the neoadjuvant setting about what those therapies are causing in terms of selection is a good thing. I'm not suggesting for a minute that it is a bad thing to give someone an effective drug in the neoadjuvant setting, because for many of the patients it will have been just the right thing to do and cured them. It's just where we've been less successful, I guess we know about it in neoadjuvant settings because we have the residual disease, and I think we should care to study that well in these repair settings.
Any question? It's okay. All right, thank you very much.
Thank you.